In this lesson, we'll explore the technical core of precision medicine, the bioinformatics workflow. The first question we ask ourselves is, what technologies have enabled the explosion of data? What formats do they use? This journey begins with the technology that triggered the data explosion, next-generation sequencing, or NGS. Let's focus on the technologies that made the explosion of biological data possible. The mother of all sequencing technologies is Sanger sequencing. In other words, the first generation of sequencing. This elegant DNA sequencing technique is based on the use of fluorescent dideoxynucleotides that interrupt strand synthesis. Strands of different lengths are then separated with extremely high resolution using capillary electrophoresis for the final reading of the nucleotide sequence. This technique, which reached its peak technological expression with the Human Genome Project, is still the gold standard for sequence validation. However, it only allows you to read one sequence at a time, which makes it inefficient for large-scale analysis. Parallelization was achieved with NGS sequencing, also known as massive parallel sequencing. Unlike Sanger, NGS uses technologies based on a massively parallelized process that analyzes millions of DNA fragments at the same time, drastically reducing time and costs, and thus allowing for the comprehensive analysis of genomes on a large scale. Second-generation technologies, such as Illumina platforms, have long been considered the standard and are based on the principle of sequencing by synthesis. The main limitation of these technologies is that they can produce sequences of up to 300 base pairs at most. Emerging third-generation technologies, such as Oxford nanopore sequencing and single-molecule real-time by PacBio, is redefining the field thanks to its ability to generate long reads. These new third-generation platforms offer higher speed and the ability to directly sequence individual DNA or RNA molecules, which is a huge advantage. But the real breakthrough is their ability to directly measure the modifications present on DNA or RNA molecules, such as DNA methylation states. This has paved the way for new ultra-fast diagnostic applications, as demonstrated by the Marling classifier, which uses methylation data to subtype patients with acute leukemia. These new tools have also proven to be essential outside the clinical field. For example, in 2022, they made it possible to generate the first complete human genome without any gaps through the telomere-to-telomere project, also known as T2T. This result was made possible thanks to the use of tools that produce long sequences, solving the problem of the repeated and low-complexity regions of our genome, which, when using short sequences, create issues with uncertain assembly. Thanks to these huge technological leaps, precision medicine is undergoing a profound transformation and could rewrite the near future. Imagine being able to go to your doctor and have your genome sequenced, or getting a metabolic profile of your blood almost in real time. The introduction of real-time sequencing isn't just an improvement. It's a technological breakthrough that enables new clinical possibilities that were previously unthinkable, like rapid diagnostics in intensive care. To manage the data produced by these technologies, it's essential to understand the basic processes and standard file formats. The Sanger method was the first high-resolution sequencing method and dominated for decades. Here, the sequence is obtained by reading the chromatogram. In NGS, on the other hand, we have sequencing by synthesis, which recreates a complementary strand to the fragment to be sequenced, allowing us to deduce the sequence. To observe a strong signal, a clonal cluster is generated in a discrete area of space, containing a large number of copies of the molecules to be sequenced. Once many clusters are obtained, that is, many clones of numerous molecules, sequencing takes place, which involves the cyclic synthesis of the complementary strand. The synthesis uses 4 fluorescently labeled DNTPs, modified so that only one nucleotide is incorporated per cycle. After incorporation, an optical system made up of a laser and a camera captures an image of the flow cell, recording the fluorescent signal, that is, the color of the DNTP incorporated by each cluster. This is followed by a wash that removes the fluorophore and reactivates the last nucleotide, making it ready for the next synthesis cycle. To convert these fluorescent signals into text, base calling is required. This is the process that translates the raw signals from the sequencer, that is, sequences of images, into a sequence of A, T, G, and C bases for each cluster. These output sequences are called reads, which are essentially readings of the sequences. The process also measures the accuracy of the call, providing a quality score associated with each individual base in the reads. Imagine the sequencer as a camera that takes pictures of chemical reactions. Base calling is the algorithm that interprets those images and translates them into a language that a computer can read. The output of this process is stored in standard formats that make collaboration and analysis between laboratories easier. While NGS excels at high-throughput sequencing, meaning it produces a lot of data, third-generation sequencing, like those offered by PacBio and Oxford Nanopore, has revolutionized the field by focusing on read length. These new technologies produce extremely long reads, called long reads, sometimes tens of thousands of bases long. This is a result that's impossible for NGS, which produces short reads of up to 300 bases. PacBio uses a real-time synthesis system, working with single DNA molecules, even without the need for amplification. In the case of Oxford Nanopore, sequencing happens by forcing the DNA molecule through a nanopore and measuring the disturbance in the ionic signal. Each combination of 5 or 3 bases causes a characteristic disturbance that is translated into a sequence. Now let's move on to understanding how the genomic data is stored. The most important file formats for sequences are FASTA and FASTQ. FASTA is a text format for describing sequences and contains a line with the name of the sequence, introduced by the greater than sign, followed by the sequence itself on the following lines. The sequence ends when it encounters the greater than sign of the next sequence, or the end of the file. FASTQ is a format derived from FASTA, developed for the outputs of sequencers. In fact, it contains the reads, that is, the raw DNA sequences, and the corresponding quality scores for each base. These scores indicate the reliability of each sequenced base, a crucial element for quality control and downstream analyses. Other fundamental formats are BAM and SAM, which, in addition to the sequences, can also contain the alignments of the reads to a reference genome or transcriptome. SAM is the human-readable format, while BAM is its complex and indexed version, optimized for bioinformatics analysis. Finally, a format that is very commonly encountered in clinical settings is VCF, or Variant Call Format. This is the standard format for describing sequence variations, that is, the data of identified genetic variants. It is one of the main result files on which clinical interpretation is based. The bioinformatics workflow begins with the management of this raw data, which presents crucial initial challenges. Once the raw data is obtained, that is, millions of fragments, for example, from 150 base pairs each, the first step to take is to perform for rigorous quality control to remove low-quality sequences or erroneous fragments. At this point, the data enters the analysis cycle that gives meaning to the fragments. Thanks to the publication of the human genome and its annotation requirements, we can map the reads to the reference genome and interpret them. If the sequencing comes from an RNA-seq, meaning transcriptome analysis, I will map the reads using specific aligners followed by quantifiers that will give me quantitative information, that is, how much a particular transcript is expressed. By using other tools, I can also obtain qualitative information, meaning which set of alternative isoforms are expressed by a gene in a given sample. In the case of DNA sequencing, it is possible to reconstruct the individual's genome using the reference genome as a guide. This process, known as assembling, is a critical step that involves reconstructing the complete genomic sequence by comparing it with the reference genome. Long reads obtained from third-generation sequencing also undergo similar processes. However, the low coverage that is often observed with this data presents unique computational challenges. This means that the development of hardware and software must go hand in hand with innovations in biochemistry to make ultra-rapid diagnostics a clinical reality.